first pioneer is named Dr. Leonard Flom. He's presently the clinical assistant professor of ophthalmology at the NYU School of Medicine. He conducts ongoing iris, ear, and biometric research of newborns and birth mothers. Dr. Flom is the 2013 inductee of the Inventors Hall of Fame for his 1987 patent entitled Iris Biometrics Technology, and is a consultant to security agencies working with the U.S. and Israeli governments on iris and skeletal biometrics. But tonight he's not going to be talking to you about any of that. He's going to be talking to you about Ebola. So I'd like to welcome Dr. Flom to the stage.
I'd like to preface this first slide, which is my unique technology to kill Ebola in vivo, in the body. I'd like to preface this introduction slide by stating the essential information that will be shown further in this presentation was sent to the heads of NIH, CDC, and World Health Organization at the height of the Ebola epidemic, the first week of August 2014. A technical officer of WHO rejected what is herein 2 weeks later. I never heard from CDC or NIH. So, second slide. It is a truism. It's far more sensible and logical to kill a killer before the killer has a chance to kill the victim. As you can see on the bottom, there have been 16,603 reported cases as of December 17th when I submitted this, with the number of deaths being 615. As of 2 days ago, the very latest figures from January 5th now stand at 20,656 cases, and the deaths now stand at 8,153. That's just in 21 days. This is a killer. Now, in order to kill Ebola, the most efficient and quickest way to do so is with ultraviolet C. As you can see in this picture, the rods are focused on above, and those rods can be removed and placed in different places. Now, in the next slide, how does Ebola kill? The virus isn't what ends up killing you. It's your own immune system. Ebola is a diabolical, extremely sophisticated virus because it surreptitiously tiptoes quietly into the immune system, and a dendritic cell, which is not the same as a nervous system, signals and essentially shuts off the ability to alert the alarm system. Now, the dormant immune system then becomes a facilitator by not attacking, thus enabling Ebola to easily infect all of the victim cells. The volume and number of Ebola-infected cells awakens the victim's cytokine protein molecules to message the human immune system, resulting in an outpouring of excessive, overwhelming number of immune bodies to attempt to kill the virus. It's like 10 different companies from a fire company coming at it at the same time. The extreme immune attack is called a cytokine storm. This storm causes the victim's cells to explode, leak blood excessively, and continuously until the victim dies. Is there a defense against the Ebola killer? The answer is yes. The best defense is always an offense. Therefore, kill the Ebola as soon as possible. How do we kill Ebola? By attacking Ebola's Achilles heel. That Achilles heel is Ebola's vulnerability to destruction by photons emanating from ultraviolet C rods, herein called UVC. Unlike viruses, vaccinia, and Lassa, Ebola is attacked and killed by UVC. The closer the UVC is placed to the Ebola virus, the more efficient is its killing power to destroy the Ebola. UVC may also destroy HIV when used up close. This is something that only technologically recently we've been able to say. Now, how do we get very, very close to the killer? We can use a kidney dialysis machine that has external tubings through which the blood circulates by means of attachment to a subject's artery and vein. By utilizing this machinery, exempting the chemicals that we use for kidney failure, the external tubing of the vein and artery can either be surrounded by photoradiation UVC rods or internalizing the rods within the tubing. With each cycle of circulating human blood, more and more Ebola virus is destroyed. This continues until the Ebola count reaches 0. The patient is free of Ebola if the count remains 0 for 72 hours. The number of cycles necessary to accomplish this is to be determined by experimentation. Now, there are three UVC, threefold proof of the benefits. UVC has been and is universally used efficiently and effectively in hospital corridors to kill Ebola, to kill bacteria and viruses at a distance. Ultraviolet germicidal irradiant or UVG kills Ebola viruses on surfaces and the air in healthcare settings. You'll excuse me if I can't read this too well because I only see really out of one eye. UVC has proven harmless to humans when used up close in circulating blood, as disclosed in numerous publications by Dr. Richard Adelson, who's a head of the Yale University's chief of dermatology, in his seminal cancerphoresis experiments. I'll show you his letter in a moment. UVC stimulates the production of human immune bodies. Now, UVC has been and is universally used efficiently and effectively and inexpensively in hospital corridors to kill bacteria and viruses. Here you see it. Their germ-killing robots are now being placed in the Republic of Liberia to aid in the battle against Ebola virus. This is in the corridors. And this is, the inventor is shown in this slide with the robot that kills the viruses and the bacteria in the corridors. UVC has been proven harmless to humans when used up close in circulating blood, as disclosed in numerous publications by Dr. Adelson. Here you see Dr. Adelson's letter to me, which I sent to him the first day that I thought of this. And he says that the idea of photoirradiating blood products with HMOP, which is sorolin, to decontaminate them is a good one. Here is, and he gives me a URL in order for me to follow up on this, but he has done more work as far as up close with UVC than anyone else. Now, UVC stimulates the production of immune bodies. As you can see here, that it has already been processed as far as inactivation and could easily be implemented in using blood banking procedures. Also, the immune bodies, the cytokine reaction is it's stopped as far as the UVC is concerned. UVC prevents the cytokine reaction. Now, in conclusion, it's an old weapon, and that's UVC photons can now be used in a new way, the same way that we use antibiotics in vivo to supplement the human immune system to kill bacteria.
Thank you very much, Dr. Flom.
A question? Dr. Flom, I appreciate that. That was fantastic, and I'm afraid to say anything about the medical nature. My questions are more on the length of time it takes to treat somebody and how we would be certain that they would be disease-free. I just didn't understand that.
That's only that can only be done as far as experimentation is concerned. We don't know how long this is take. This has never been done before, so we don't know how long it's going to take. But the chances are that if it works, it's going to work within 1 or 2 cycles.
How long does a cycle take generally if I compared it to...
Cycles take approximately a minute or 2
Okay.
to flow through. Any other questions?
Yes, thank you again so much for being here. Really enjoyed listening to your presentation. My question is more along the lines of implementation. Just focusing specifically on Ebola, it's my understanding, and again, I do not have a medical degree, so feel free to correct me, but that we've been relatively successful in combating Ebola in the United States and other developed nations. The problem really exists in developing nations in Africa, and so the lack of infrastructure has tremendously played into why the disease continues to proliferate. So I guess that's my question. How would you deploy UVC in terms of the dialysis machines and all the infrastructure that's necessary in order to effectively treat patients?
The treatment would be by exposing volunteers to, they would be attached to a dialysis machine, and the UVC rods would either be inserted within the tubing or surrounding the tubing. And then with each circulation, more and more of the viruses would be killed.
Yes, but in terms of the current sort of resources that developing nations have in terms of their hospitals, is that something that exists already, or is that something that they would need to build? And
they would have to be able to build this in Liberia. There was a very well-known philanthropist in Liberia who was sick, and he donated a large number of the dialysis units to Liberia. So if this was going to be implemented, it would be done in Liberia.
Is it as simple as just putting the rods near the tube where the blood's
Exactly,
flowing?
yeah. I've spoken to the people who make the rods, and they say that this is a very, very interesting concept, and they would like to do this in Liberia because they already have the robot there. Any other questions?
I guess less of a question, more of a comment. Again, given where I come from, I'm much more focused on connectivity and information. You might want to, at least in the back of your mind, think how to, in order to encourage mass adoption, information is always very important to moving things forward, right? Especially knowing more and more about a certain process or technology. So again, more of a comment than a question is to think about how this process, specifically what data points and metrics you can measure in the process while you're trialing it and actually executing it. What information is key to extract from that with the, as Internet of Things and other technologies start to evolve, what information is key to extract in order to better tune the process and feed that information back into other information
Well,
sources.
obviously, I'm an ophthalmologist, and my area of expertise is ophthalmology as well as biometrics. I can't answer your question. Only virologists would be able to give you that answer if they were willing to experiment with this concept.
Understood. It was, like I said, it was more of a suggestion than it was a question.
Suggestion as to how this should be done?
As to just keep in mind as you move forward with trying to move this moonshoot forward, what key information points are going to be critical in order for the success of it, so it encourages mass adoption?
I would have to rely on Google in order to do this.
Done.
Because I can't do this, and the CDC and the WHO, who knew of my credentials, they were asking me to perform this. I don't have access to chimpanzees, and I don't have access, and certainly don't want Ebola. But
I
this
can
is
give you
a
one of my
suggestion.
chimps if you'd like. I can give you one of mine as well. I guess we'll end it with how you started the presentation. You said that you presented this information to the CDC and others, and no one acted. Why do you think they didn't act on
That's
that?
a very good question. I can't answer that for you, but I can tell you this, that if I can read to you what I got from the people at WHO, I understand that there is evidence that the effect of UVC on Ebola, and that there is safety data on the use of the UVC humans. However, the current WHO guidelines on required evidence of consideration of treatments on Ebola require that the treatment has efficacy in animal models. It's ridiculous. This is ingenuous, too, because I already presented the fact that this has already been available. Why would they ask me to do this? They can do this. If they were really interested in this, they have the animals and they have the Ebola. They could have done this.
So we'll continue this animal conversation later tonight. Thank you again for your time.
You're welcome.
Appreciate it.
Thank you very much, Dr. Flom.